Okay. Welcome to today's lecture. Today's lecture will not be too long. We are going to be looking at general biochemistry, and we are going to be focusing on the nucleic acids. If you remember last semester, we were actually looking at amino acids and the chemistry of amino acid. So we are going to actually look at the nucleic acid. And we are not taking the full lecture on nucleic acid. We are just going to be looking at the hydrolysis of nucleic acid and also nucleic acid protein complexes. So we are going to start this lecture by looking at the objectives of this lecture. And I'm saying that by the end of this lecture, the student that is listening to me, will understand the following. 1. How nucleic acids are being hydrolyzed. 2. The product of the hydrolysis of nucleic acid. The difference between acid and alkaline hydrolysis of nucleic acid. And we are going to look at what nucleic acid protein complexes are, and we're going to look at their functions. Now, but before we go into this, I would have loved us to have a brief online quiz on this subject of nucleic acid. One, by now you would have known from the previous lectures by Ogunwa that nucleic acids are polymers of nucleotides. And there are two types of nucleic acids. I know he have taught you about the RNA and the DNA, their components. RNA meaning ribonucleic acid, DNA meaning deoxyribonucleic acid. Now these acids are just chains of nucleotides. Now what is a nucleotide? I'm just trying to do a brief rundown of what you have known. A nucleotide is normally made up of a nitrogenous base, a sugar moiety, and a phosphate. That is nucleotide. Now for nucleosides, it's just a nucleotide without a phosphate. So the component of the nucleoside is only the sugar base and the nitrogenous base. Now, for this nucleotide or nucleic acid, the sugar base is always a pentose sugar, and this pentose sugar can be of two types. You have the ribose sugar and you have the deoxyribose sugar. This deoxy means that at the position 2, the carbon 2 of the sugar moiety does not contain a hydroxy group, so it is deoxy, and it makes a whole lot of difference between the RNA and the DNA. I'm not going to go back to that lecture, but I will advise you go back and do the needful. So, how are nucleic acids hydrolyzed? The first important thing is knowing what is hydrolysis. And we are saying that hydrolysis is the breaking of bonds in molecules using water. Breaking of bonds in the molecules using water. It is not the breaking of water molecules. You are breaking a bond and you're using water to do the breaking. That is hydrolysis, just as the name implies. Hydro and lysis. Hydro meaning water, lysis meaning cutting. So it is the cutting of bonds, breaking down of bonds using water molecules. Now, in nucleic acid, when you look at the structure of the nucleotide or the nucleic acid, we find out that there are sites where this breaking takes place. One of the sites can be broken in the connection between the sugar moiety and the nitrogenous base, or it can be broken at the phosphate backbone, or it can also be broken even in the base. So the product of this hydrolysis will always come to be either the purine or the pyrimidine base, the oligonucleotide or nucleoside, and the ribose or the ribose sugar and a phosphate. So it is just hydrolysis is just this, breaking all the segment, all the things that makes the nucleic acid to come together. Now, nucleic acid polymers can be extremely long and therefore difficult to manipulate, and sometimes even very difficult to sequence. Similarly, it may be desirable to arrange them in regions within the polynucleotide, where these regions could be a place you insert or a place you delete, and all those and all that. When you begin to look at mutations, we begin to look at these insertions and these deletions. So, but the things we want you to understand at this point is that nucleic acid contains regions, and these regions can be broken and they can be inserted. Now, all this process requires that the phosphodiester bond have to be broken, okay, for it to actually happen, whether insertion, whether deleting, whether DNA repair and all those things. There must be a hydrolysis of the phosphodiester bond, and this bond is the bond within the polynucleotide molecules. Now, this hydrolysis could either be done with an acid or with an alkaline or base, or it can be done with an enzyme. So let's look at the types of hydrolysis in nucleic acid or of nucleic acid. The first one we'll be considering is the alkaline or base catalyzed hydrolysis. This type of hydrolysis occur in RNA, and the reason they occur only in RNA is because the RNA has a hydroxy group at the position 2 and at the position 3 of the sugar moiety or the ribose sugar. So this OH at the 2' and 3' positions helps to actually enhance this hydrolysis process. So the hydroxyl ion assists in a nucleophilic attack on the position 2, that's the C2' hydroxy group on the phosphor, thereby yielding an intermediate, and this intermediate is called cyclic 2,3 phosphonucleoside. Remember how the name goes: cyclic 2,3 2', 3' phosphonucleosides, not phosphonucleotide. Okay, phosphonucleoside. It is actually a nucleotide that is formed, a cyclic nucleotide that is formed. But because you have mentioned phospho, you don't call it nucleotide again. I guess we are making it clear now. And this cyclic 2', 3' phosphonucleosides are very, very unstable. In other words, it can easily be hydrolyzed into nucleoside 2' phosphate or nucleoside 3' phosphate. We see that in the next figure. Now, this type of hydrolysis occurs when the pH is above 7. That is why it is alkaline in nature. The DNA molecule do not undergo alkaline hydrolysis because of the absence of the hydroxyl group at position 2 of the ribose sugar. Now, this is where most students always get it wrong. They will say it is because of the absence of oxygen at the position 2'. No, it is not the absence of oxygen. It is the absence of a hydroxyl group. Okay? Now, let's look at what is happening here. Now, you see the base. what you have at the left side is the chain of RNA, okay? And this RNA is being hydrolyzed using a base, and it will form the cyclic 2' 3' phosphonucleoside. Now remember I told you that these cyclic 2' 3' phosphonucleosides are unstable, so it can easily be hydrolyzed by water to form nucleoside 3' phosphate, or most of the time some people or some books will have it as 3' nucleotide, okay, but it's generally known as nucleoside 3 phosphate, and then the 2' component, which is the nucleoside 2 phosphate. So now these two are the product of alkaline phosphate. Now these reactions can still go on to break these nucleosides and nucleotide and 2' and 3' phosphates. It can go down to break it into their base, into the sugar, and into the phosphate group, depending on the treatment. Now the next one is acid-catalyzed hydrolysis. Now, acid hydrolysis causes the depurination of nucleic acid, which means it can occur in DNA and RNA. Now, basically, what acid hydrolysis does is to remove the purine from the nucleic acid. Purine, for instance, the adenine and the guanidine. And the guanine, not guanidine. The adenine and the guanine. Now, what does the acid do? The acid attacks the N-glycosyl bond. The N-glycosyl bond. In the case of purine, it is the 1N, the N-glycosyl bond. In the case of pyrimidine, it is the 9N glycosyl bond, which it does to release the adenine or the guanine or the base, depending on it. And when it removes the base or the nitrogenous base, it will leave a site, it will leave that place vacant, and that place is now known as the apurinic site. Okay, now sometimes the phosphodiester bond may be broken, and much harsher treatment is required when you're dealing with the removal of pyrimidine. Okay, so the N-glycoside, to remove the pyrimidine N-glycoside, but this also follows the same process as we are going to see in this structure. Now here you can see the left side where you are having the nucleic acid, whether DNA or RNA, and it is being treated by an acid. Okay, we can see the attack where this base is being removed, and when this base is being removed, you will have this structure without the base, and this structure where the base is missing, we declare it, or we say it is an apurinic site. Okay, now with this you find out that the nitrogenous base have been removed from the DNA or RNA, can be removed from the the nucleic acid. Now in the presence of water, the reaction continues, and it will break it down into the phosphate group and also the sugar group. So it's somehow dismembered the nucleic acid. Now we also have enzyme hydrolysis. Don't forget that enzymes are organic catalysts that helps in speeding up reactions. Okay, now I will always have us remember that enzymes, even though we call it organic catalysts, are not inorganic in nature. They are all organic, and enzymes partake in the reaction but returns to its native state after the reaction. So acid hydrolysis, which is enzyme hydrolysis, in which the enzyme is used in this hydrolysis to hydrolyze nucleic acid. Now, the enzyme that hydrolyze nucleic acids are generally known as nucleases. Okay, they are required for the normal housekeeping function in the biosynthesis and turnover of nucleic acid. So you will always see nucleases in both the production, the synthesis, the breakdown of nucleic acid. Now, they are required actually in the breaking down, in the digestion of nucleic acid. There are two general types of nucleases. You have the exonucleases and you have the endonucleases. The exonucleases starts, hydrolysis from one end of the polynucleotide. Either it starts from the 3' or the 5' end, depending on how specific the enzyme is. Enzymes are really specific in their function. And exonucleases break this thing down sequentially. In other words, it will break A before B, before C, before D, or depending on how you're numbering the nucleotide sequences. Now, what is it hydrolyzing? Is the phosphodiester bond. Is the phosphodiester bond. That is what it actually breaks down. So for enzymatic hydrolysis, it does not break down, remove the purine. Okay. Now, the second one is the endonucleases. As the name implies, endo, it means that the cleavage is in an internal site within the polynucleotide sequence. Although there might be multiple internal sites they are susceptible to, endonucleases do not sequentially hydrolyze the phosphodiester bond. Okay, remember, exonucleases, we do it sequentially. After the first one, it will now remove the second one, and so on, depending on where it is coming from. But endonucleases, it breaks internally, and therefore it does not consider the sequence that is breaking, even though it must see a particular sequence to recognize where it's going to bind before the cutting. Now, there are also two types of endonucleases. The two types of endonucleases, depending on the either side of the phosphodiester bond. For instance, we have the type A nucleases. The type A nucleases cleaves on the 3' side of the phosphodiester bond. I will show you that in just a moment, such that it leaves the phosphate on the 5' end, or the side of the polynucleotide. So when it is breaking, it breaks on the 3' side, leaving the phosphate attached to the 5' end, while the B nucleases cleaves at the 5' end of the phosphodiester bond, therefore leaving the phosphate attached or remaining on the 3'. Now, what this is what we mean. When this type A endonucleases is attacking the nucleic acid, it is going to break these phosphodiester. For instance, there are two bonds, ester bonds. This is the first one, and then we have the second one. Okay. Now, when type A nucleases attack, they break it in such a way that the phosphate group will remain to the 5' end, while the B type will break it in a way that the phosphate group will be attached to the 3' end. I guess I'm making a little sense. Now, types of endonucleases. Endonucleases, like we said, are enzymes that cleaves the phosphodiester bond within the nucleic acid chain. There are different types. classified based on the structure, the substrate specificity, and the mode of action. So let's look at some of them. We have the restriction endonucleases. And I said these enzymes recognize specific DNA sequences and cleave the DNA or near the recognition site. This type of restriction endonucleases are widely used in molecular biology for DNA manipulation and genetic engineering. So restriction endonucleases are sometimes called restriction enzymes, and are enzymes that code DNA at specific recognition sequences. That was specific recognition sequences, which we are going to say that it must recognize a particular sequence in the DNA before it can cut, otherwise it will not hydrolyze. It must recognize a sequence. For instance, the EcoR1, this enzyme recognizes and codes DNA at the sequence GAA-TTC. Now, what GAA-TTC means, that if it recognizes a sequence, and that sequence is guanine, adenine, thymine, cytosine, it will cut. And it produces a sticky end with a 5' handoff. In other words, from the 5', it will stick out. Okay? It will stick out, which can anneal to another piece of DNA cut with the same enzyme. So it can cut and still join together when it sees another DNA. So whenever it cuts, there is an opportunity to insert a DNA within that region that is cut, and it will still seal it again. We have the hint3. Now this recognizes the sequence AAG-CTT. AAG-CTT. Remember, EcoR1 recognizes the sequence GAA-TTC, but this one recognizes AAG-CTT. Okay? And it produces a blunt end, which is not complementary to the other. But the other one, the first one we talk about this, they must be complementary, okay? And cannot align. Now we have the BAMH1, we have the NOT1, and so on and so forth. Now another kind of endonuclease is the DNA repair endonucleases. There are endonucleases that are involved in the repair of DNA and the correction of DNA sequences, maybe after replication processes. So we say that these enzymes are involved in repairing damaged DNA by cutting the damaged strand, which is then repaired by DNA polymerase and ligases. There are several types of DNA repair endonucleases, and these endonucleases include the base insertion repair, nucleotide insertion and repair. We have the mismatch repair. We have the homologous recombination endonucleases, and we have the non-homologous end joining endonucleases. So these ones are involved in DNA repairs. Now we have the homing endonucleases. These are found in introns or intern and cleaves DNA at specific sites to promote their own mobility. Homing endonucleases, also known as homing endonucleases gene, are enzymes that recognizes and cleaves specific DNA sequences. There are some examples of this which we might not be able, just take note of it. You have the IP-pole. This enzyme is found in the mitochondria of yeast and recognizes and cleaves DNA sequences of 15 base pairs. Okay, you have the I cell, and so on and so forth. It's just for you to note them. When you come into biochemistry proper, you will begin to see majority of these things. Now, it is not only DNA that endonucleases are involved in. There are some that are involved in RNA processing, and some of those are, we just mentioned it, the ribonuclease P, the RNase, the dicer, the endonuclease V, and so on and so forth. We also have the ones you find in the mitochondria. Remember that there are, apart from the nucleus, the mitochondria and the protoplasm and the chloroplast are two places where you can find the DNA. Okay, so in these places we have endonucleases that helps in one function or the other. So we have the mitochondrial endonuclease, you have the endonuclease G, the homing endonucleases, the DNA cell gamma, the XPG, and so on and so forth. The mitoribonuclease, they are all part of it. Now we have the micrococcal nuclease, and we say that this is a non-specific nuclease that leaves both single-stranded and double-stranded DNA and RNA. Now we have the Cas endonucleases. Now, Cas endonucleases are very much in use in today's research because they are normally used to, cut the DNA, and it makes, at this time, you're the one that is choosing where to cut. Okay, so we say that they are found used in the CRISPR-Cas system, which provides defense mechanism against invading nucleic acids such as viruses and plasmids. And may I add here that the mechanism of CRISPR has offered so much, promises in treating some diseases. For instance, sickle cell can be treated with CRISPR system, where you will actually go directly to remove the particular gene or gene that is actually responsible for the problem. So those are also endonucleases. Now, we've talked about exonucleases. Remember, we say they cut from one side or the other. So what are the types of endonucleases? We have the 5' to 3' endonucleases. As the name is, you've already known what it should be doing. It's cutting the DNA sequence or the RNA sequence from the 5' end to the 3' end. An example includes exonuclease 1 and exonuclease 3. We equally have the 3' to 5' endonucleases, and just as we defined before, it helps to cut the nucleotide from the 3' of the DNA or RNA to the 5'. An example is the endonuclease 2 and the exonuclease 2 and exonuclease T. Now, endonucleases, we have the one that cleaves the internal phosphate bond, of some nucleases, and you just call them exonucleases. Example is S1 nuclease, the Bio31 exonuclease. You also have example of exosomes. These are complex proteins. They are not they are not nucleosides per se, but they are complex proteins. They complex actually with exonucleases to degrade RNA and so on and so forth. You have the RNA-H. This one you'll always see it in kits that are used for DNA analysis. Now, let's quickly look at nucleic acid and protein complexes. By definition, the nucleic acid protein complexes are structures formed by the association between nucleic acids such as DNA, RNA, with proteins. These complexes play essential roles in a variety of biological processes, which including the gene expression, the DNA replication and repair, transcription, translation, and protein synthesis. You will get to know about all these big words when the time comes. Now, the nucleic acid protein complexes are formed by the interaction between nucleic acid and protein component. The nucleic acid component provides the template or the information necessary for the protein to perform its function. So while the protein component contributes the enzymatic or regulatory activity necessary to the process. So you see there is these two complexes. Each component has what it's doing, okay? The information is stored in the nucleic acid, while the enzymatic process is found within the protein complex. Now, examples of this nucleic acid protein complex, there are many of them, but we are going to take few examples, and they are, you have the DNA polymerase, okay, you have the ribosomes, you have the transitional factors, and so on and so forth. In the chromatin also is a complex, and then the telomerase is also the complex. So you just have to go through it. For instance, the telomerase is an enzyme that adds short repeated sequences at the end of a chromosome, called telomere. Now, telomerase is composed of both both RNA and protein components, and forms a complex with DNA at the end of the chromosome to maintain its integrity. What are the functions of these nucleic acid protein complexes? One, they are involved in gene expression regulation. Okay, they are involved in DNA replication and repairs. They are involved in DNA processing or RNA processing per se. The nucleic acid complex that are involved in RNA splicing, editing, and transport, which are necessary for proper functioning of the RNA molecules. Now you have the ribosomes, okay, you have the chromosomal structures, you have the cell signaling, and you have immune responses. All this is where the nucleic acid protein complexes can function. So, if in summarizing this, we can say that the primary function of nucleic acid protein complexes is to store and transmit genetic information. This complex mediates the transfer of genetic information from DNA to RNA, and then through protein synthesis. Okay, protein synthesis to functional protein. Now, the interaction between the nucleic acid and the proteins are essential for various cellular processes, which include DNA. We mentioned this before. This cellular process includes DNA replication, transcription, translation, and DNA repairs. In addition, their roles in the genetic information transfer. Nuclear proteins also play a crucial role in gene regulation. The regulatory proteins bind to specific DNA sequences and control the expression of genes by either promoting or inhibiting transcription. Overall, the formation of nucleic acid protein complex is crucial to maintain cellular homeostasis through the precise regulation of gene expression. So, an outline of this function of what we have mentioned is this. If you look at the outline, we say, the outline 1. Function of this complex is that they storage and transmission of genetic information. And how do they do it? Either they mediate the transfer of information from DNA to RNA, so they facilitate protein synthesis. Okay. Now, they regulate gene expression by binding to regulatory proteins as specific DNA sequences. They control gene expression through promoting or inhibition of transcription, and they are essential in cellular processes such as DNA replication, transcription, translation, and DNA repairs. Okay, and then I will stop here to look at your questions, and then you wait.